Hello everybody. Welcome back to NeuroMD, and today we're going to talk about the anatomy and physiology in neurology. So in previous videos, you've noticed that we were talking a lot about the pathology of the CNS, and also mentioned simple basic structures of the brain that you need to know. Let's further dive into the further details. What else do we have in CNS? We have neurons. You might heard of them. If you haven't, let me tell you. Neurons are basically signal transmitting cells of the nervous systems. If you're thinking, or if you want to feel a pain stimulus or anything that is associated with your brain doing something, your neurons are the ones who transmit signal from one neuron to another to send a signal. Consider them as a messenger of the brain. Now, let's get into it. Signal transmitting cells of the nervous system is given this. It sends signals to transmit the message. They are permanent cells and they do not divide into adulthood. As you know, if you get injured, your cells divide to heal your wound. But here in neurons, they are permanent cells and they never divide. That means if they get damaged, it's a permanent damage. You can't do much about it. They are signal relaying cells with dendrites, receive input. Now they have dendrites, the structure of the neuron, and cell bodies and axons. Axons are something that send output. So dendrites will receive the input. For example, I want to send a message to my arm. So I'll send my message to through dendrites, and dendrites will then through the cell body send the message to the axons. Axons will go to my arm and tell my arm whatever I need. So this is how it goes. You need to remember dendrites, cell bodies, and axons. Pretty simple. Neurons just consist of three structures. Cell bodies and dendrites can be seen on Nissl stains, RER. RER is not present in the axons, so this is more of a histology stuff. If you're working in lab as a lab technician, you would be able to see the cell bodies and dendrites, but for axons, you can't see them with this staining. Neurons have astrocytes. Astrocytes are most common glial cell types in CNS. Physical support, repair, extracellular, K+ buffer, removal of excess neurotransmitter, component of blood-brain barrier. Remember this part: glycogen, fuel reserve buffer, reactive gliosis in response to neural injury. Basically, the thing about astrocytes and all this neuron and anatomy and physiology is that you just need to be familiar with all these things. You don't need to cram. You don't need to, really waste your time on these, but just know what they are and be familiar. So when we talk about disease progression and pathology, you would know which structures are we talking. This will help you map out that which part of the brain and which structure in the brain and what are we talking about. So it's just to help you map out CNS. Astrocytes derive from the neuroactism. Astrocyte marker is GFAP. So this is all about astrocytes, and this is how it looks like. Now, move on to microglia. Microglia are phagocytic scavenger cells of CNS. Remember that they are the phagocytic cells. Mesodermal mononuclear origin, activation in response to tissue damage. So if there is a tissue damage, then these will be activated. Release of inflammatory mediators. What are inflammatory mediators? Nitric oxide or glutamate, things like these. Not readily discernible by Nissl stain. Again, can be seen. Now, HIV infected microglia fuse to form multinucleated giant cells in CNS. Now, why you have to know this? Because in future, when we are talking about diseases, you would know that what microglia cells are and what's their function. Just remember they are phagocytic scavenger cells, which release which are inflammatory mediators. For example, if there's a tissue damage, anything happens, they will release all these things. Okay, this one. Ependymal cells, ciliated simple columnar glial cell line, the ventricles, and the central canal of spinal cord. Spinal cord anatomy, apical surfaces are colored in cilia which circulate CSF. CSF is our cerebrospinal fluid, and microvilli which help in CSF absorption. Okay, now specialized ependymal cells, choroid plexus, produce CSF. So this is not just the anatomy, but we are also mentioning the physiology here. Now move on to myelin. They increase the conduction velocity of signals transmitted down axons. Now what are axons? Remember, they send out the message. Send out the message how? Through the signals. Think about it as the electric signals being transmitted, right? So myelin is something that helps increase the conduction velocity, increase the speed of signals transmitted down the axon. You know how sometimes even as adults, if we touch something and it's very, very hot, sometimes we're not really paying attention, and it kind of takes some time to have that reflex that, oh, it's hot, remove your hand from it. So that's the conduction velocity. Sometimes it's slow. That's why we need myelin to increase the conduction velocity. Now, a saltatory conduction of action potential at the rods of Ranvier. This is what we've talked a lot about in high school, where there are high concentration of sodium channels in CNS, including CN2. Myelin is synthesized by oligodendrocytes. In PNS, which is peripheral nervous system, including cranial nerve 3 and 7, 3 and 12. Myelin is synthesized by Schwann cells. So in CNS, we need myelin, and you see myelin is synthesized by Schwann cells, which is happening in PNS, which includes the nerve C and cranial nerve 3 and 12. But in CNS, that includes including cranial nerve 2, myelin is synthesized by oligodendrocytes. So that's also something you should be familiar with, and do a lot of Anki flashcards. It will help you remember and retain all the information. So it wraps and insulates axons, which send out signals again. Now, increase phase constant and increase the conduction velocity. Remember, this is the picture right here. You can see everything very clearly. COPS, CNS is oligodendrocyte, CNS, C is for CNS, O is for oligodendrocyte, PNS equals to Schwann cells. So P stands for PNS and S stands for Schwann cells, so COPS. Remember this, CNS, oligodendrocytes are responsible for producing myelin, and in PNS, Schwann cells will produce myelin desynthesize. Okay, so COPS is the mnemonic. Now let's get into Schwann cells. What are Schwann cells? Schwann cells promote axonal regeneration derived from neural crest. Each Schwann cell myelinates only one PNS axon injured in Guillain-Barré syndrome. So this is, you could see Schwann cell, the myelin sheath is right here, this is the nucleus, and these are the rods of Ranvier. Nodes of Ranvier are basically the area on the neuron which is not covered with myelin. So this would make the signal to jump from one myelin to another and skip the nodes of Ranvier. Now oligodendrocytes actually are This is a picture of the cells that produce myelin for CNS. Myelinate axons of neurons in CNS. Each oligodendrocyte can myelinate many axons. Predominant type of glial cells in white matter. They are basically in white matter, okay? Derived from neuroectoderm, fried egg appearance histologically. So in histology, you would see them and you would think of it as, oh, that's a fried egg. Yo, I'm hungry. But it's actually this. Injured in multiple sclerosis. So if there's a patient with multiple sclerosis, they won't have oligodendrocytes to produce myelin, and that would decrease the conduction velocity. And progressive multifocal leukoencephalopathy, VML, which is very common, and leukodystrophy. So these are the three conditions where oligodendrocytes will be damaged and won't be produce any myelin. They won't be myelinating any axons, which indeed decrease the conduction velocity. And remember, Schwann cells are for PNS. They work for PNS, and Schwann cells are damaged in Guillain-Barré syndrome, which is again very common, and it again decreases the conduction velocity. Now we have sensory receptors. Sensory receptors are quite a lot. Now let's go through them one by one. We have free nerve endings, Wisner corpuscles, Pacinian corpuscles, Merkel-Dis, and Rufinimo corpuscles. So sensory neuron fiber type, their location, the senses. We'll get into this later. Let's talk about peripheral nerve. We're here to talk about peripheral nerve. This is how they look. They are basically endoneurium, thin, supportive connective tissue that enseeds and supports individual myelinated nerve fibers. Perineurium, blood nerve permeability barriers around the vesicle of nerve fibers must be rejoined in microsurgery for limb attachment. So when there's a robotic microsurgery, they focus a lot about rejoining them. And epineurium, dense connective tissue that surrounds entire nerve vesicles and blood vessels. So these are the peripheral nerves. Of course, they're everywhere except the brain. Now let's go through the terms. Endo means inner, peri means around, epi means outer. That's all for peripheral nerve. These are the topics that you should go by real quick. They don't spend a lot of time here. Don't get lost. Focus on diseases, a lot on diseases, diagnosis, and treatments. Next one is chromatolysis, the reaction of neuronal cell body to axonal injury. Now remember, neurons get damaged, their damage is permanent, and the reaction is chromatolysis. Changes reflect what are the changes? Increased protein synthesis in effort to repair damaged axon, characterized by there would be some swelling. You could see here the round cells are swelling and the displacement of nucleus to the periphery, and you could see the nucleus, it's being displaced to the periphery, dispensation of missile substance throughout the cytoplasm. So it's basically the damage that's happening. And in this degeneration, disintegration of axon and myelin sheath distal to site of axonal injury with macrophages removing debris. So proximal to the injury, the axon retracts and the cell body sprouts new protrusion that grow toward other neurons for potential re-innervation. Serves as preparation of axonal regeneration and functional recovery. So again, this is not very important to be going in details. Yeah, in the previous videos I promised that I would definitely talk about cerebellum. So here we are. It modulates the movement, aids in coordination. We discussed this before. Cerebellum, whenever you hear it, just think of balance. Input, contralateral cortex via middle cerebellar peduncle. Cerebellum is lying right here, and ipsilateral proprioceptive information via inferior cerebral peduncle from spinal cord. The only output of cerebellar cortex is Purkinje cells, and always inhibitory deep nuclei of cerebellum contralateral cortex via superior cerebellar peduncle. Deep nuclei is lateral to medial. Dentate, don't eat greasy food because this is the mnemonic as dentate, emboliform, globose, and vestigial. Don't eat greasy food. That's how you remember. Just be familiar with all these structures and their physiology to help better understand all the diseases that exist. Lateral lesions affect voluntary movement of extremities. Lateral lesions, remember, what will they affect? They will affect voluntary movement of extremities that you do on purpose. When injured, propensity to fall toward injured ipsilateral side, and medial lesions like at the vermis, vestigial nuclei, or here the truncal-ataxia wide-based cerebellar gait. This is a specific kind of gait that happens. The stigmas, head tilting, generally result in bilateral motor deficits affecting axial and proximal limb musculature. These are the medial structures. That's all for cerebellum, and in this one we are just gonna be very specific on what we are gonna discuss. So this one is content that we will talk about later. The next thing I wanted to discuss was the ventricular system, which we recently talked about. Here we go. Let's review the ventricles we have. The 4 ventricles. Lateral ventricle 1, 2, and the third ventricle right here, and the fourth ventricle over here. And you can see all these horns because they actually look like horns, so you can tell. Lateral ventricle to third ventricle, we are right and left interventricle foramina of Monroe. Third ventricle to fourth ventricle via cerebral aqueduct of Sylvius. Fourth ventricle to subarachnoid space via foramina of Luschka. Lateral, because L and L. Foramen of Mesenteric medial. Remembering is the key, and to remember stuff you have to make mnemonics here, otherwise you're just gonna forget. CSF made by choroid plexus, located in lateral and fourth ventricles. So here we go. Travels to subarachnoid space. Remember where the subarachnoid space is, and we have foramina of Luschka and mergente ribs of biarachnoid granulations, and then drains into dural venous sinuses. So this is our ventricular system, and all these things, just be familiar with this system and from where to where it goes, and that's all you need to know. Don't go in details. And now we will mention some about brainstem. Ventral view. This is the ventral view of the brainstem. So what are we looking at? We have pons right here, and here's a list of all the cranial nerves. So 4 cranial nerves are above pons, which is 1, 2, 3, 4. Let's see where our pons is. Let's point out. Where is my marker? So this is pons, and they say that 4 cranial nerves are above pons. So cranial nerve 1, 2, 3, 4. Olfactory bulb, olfactory tract, optic tract, and fourth arises dorsally and immediately. So look at this, 1, 2, 3, and fourth one is here, and we respond pons is right here. So all the four nerves are above pons. Next, for cranial nerve, exit the pons, which is 5, 6, 7, 8. Look at that. Pons is right here. This is cranial nerve 5. It's exiting the pons. 6, 7, 8. It stops right about here. Okay, and next four cranial nerves are in medulla, 9, 10, 11, 12. Now let's look at 9, 10, 11, 12. They're right here, and they are in the medulla, which we can clearly see right here. And remember, this is a ventral view of the brainstem, not anterior. Now, nuclei are medial for central cranial nerve. Nuclei are medial 3, 4, 5, 6, and 12. Factors of 12 except 1 and 2. So that's about the ventral view. Now let's move on to the dorsal view of our brainstem. Look at that. This one is much clearer, I believe. It gives you perspective, you could say. So I'm just testing my pen, but I lost the actual pen I want. Yeah, this one. Dorsal view, cerebellum removed. Poor cerebellum, they didn't mention here. Now let's look at our ventricles. Third ventricle right here. And then we have glands right below the third ventricle. The medulla is right here. And then we have a lot of other structures, but don't forget thalamus. Let's move on to pineal gland, which is also in the brainstem, and you could see clearly in the dorsal view. What's the function of pineal gland? Right, melatonin secretion and circadian rhythms. What are they? Your sleep cycles. Now, direct eye movement to stimuli, noise, movements, or objects of interest. It's all about this one. Oopsies. Okay, the inferior one is for auditory, and your eyes are above your ears, of course. So, and the superior colliculus visual is above the inferior colliculus, which is auditory. So, because your eyes are above your ears, that's why superior colliculi is the one that does the eye movement, so direct the eye movement to stimuli like noise and movements or objects of interest, and your ears are under your eyes, so they're below your eyes, so inferior. Hence, inferior colliculi is responsible for auditory stuff. And that's all for the pineal gland and other structures. Now let's move on to our next topic, which is cranial nerve nuclei. Now, if you pay some attention here, you would see that cranial nerve nuclei is located in tegmentum portion of brainstem between dorsal and ventricle portions. We just went through the dorsal view and the ventricle view. So, back to cranial nerve nuclei. You have midbrain, pons, medulla, and spinal cord. Okay, so in midbrain you will see nuclei of cranial nerve 3 and 4. Pons, you will see the nuclei of cranial nerve 5, 6, 7, 8. Medulla, nuclei of cranial nerve 9, 10, 12. And spinal cord, you would see the nucleus of cranial nerve 11. So this is the whole map, and lateral nuclei equals to sensory, alveolar palate, sulcus limitans, medial nuclei, motor, basal plate. This is the picture to clearly visualize everything. So cranial nerve and the vessel pathways. In this picture, you can localize all the cranial nerve from 1 to 12, and you can see the arteries and the veins. Let's point out some of the structures for you. Where is my pen? I keep losing my pen. So, the cranial nerves you can see clearly for ophthalmic artery. The artery that is for your eyes is right here, and the middle meningeal artery is right this one, and then it brings us to jugular vein, which is right here. Okay, this is all about brainstem, the cranial nerves, and the vessel pathways. And I believe for today, you should just take it lightly and not go in details of all these nerves and pathways. There is a new video coming up which is gonna give you a very clear perspective of all the cranial nerves and their pathways and other tracts of the spinal cord, and more details, clear explanation on the brainstem. This one is just for you to be familiar with everything, and it's for you to get ready for the next video where I'm going to talk a lot about the structures, and this will give you a perspective. Oh, I've heard neurons, I've heard about neurons, I've heard about cranial nerves, I've heard about the pathways and tracts. So yeah, that's all about it, and that's all for today. Thank you very much, and see you next time. Bye-bye.